Investigating populations. There are many ways to investigate populations, and you need to ensure your samples are random, you've carried out a risk assessment, and you've considered the ethics involved. So first, a look at the terms abundance and distribution. So investigating populations includes looking at the abundance and distribution of species in a specific area. Abundance is the number of individuals of one species in a particular area, e.g. the population size. Abundance can be estimated by merely counting the number of individuals in the samples taken. There are additional measures of abundance that can also be used. So frequency is the number of samples a species is recorded in, e.g. 70% of samples. Percentage cover, so non-motile or slow-moving species is like the only ones you would use percentage cover for. So this is how much of the area you're investigating that is covered by a species. Distribution is where a particular species is within the area you're investigating. So now looking at random sampling. So most of the time it would be excessively time consuming to measure the abundance or population size and the distribution of each species present in the entire area you're investigating. So instead you take samples. So you choose an area to be sampled, a little area within the area being investigated, and the sample should be random to avoid bias. So you could use a random number generator and use those as coordinates on a grid. So you'd use a suitable technique to take a sample of the population. Then you repeat the process, taking a large number of samples, and this will reduce the likelihood that your results are down to chance. The number of individuals for the whole area can then be estimated, taking the average of the data collected in each sample and multiplying by the size of the whole area. The percentage cover for the whole area can then be estimated by taking the mean of all the samples. So if you're investigating populations in a field, you can divide it into a grid. It's a bit like a large graph on the floor. And then you would use a random number generator to select coordinates. And this will give you coordinates at random. Then you will just take your samples from these coordinates. So for instance, for this one, you'd walk 15 on the meters on the x-axis, 19 meters on the y-axis, and then you'd drop your quadrat where that meets. Just a little word about running means. So it's important that you take a large number of samples to give a good estimate. And one way you can do this is to take a running mean. And this is where you calculate the mean of all the data each time you collect a new sample. And once the mean no longer changes by a large amount, so it should level off if you were to plot it on a graph, you should have data that gives a representative estimate for the whole area. So there are many different methods for studying populations of organisms, but you must choose the most suitable one to use. And this depends on the types of organisms and its habitats. So quadrats and transects can be used for studying immobile organisms like plants and corals, or slow-moving organisms like limpets. On the other hand, if you're studying more movable motile organisms like insects, like insects or small animals like mice, nets or traps would be more useful. So a quadrat is a square frame which is usually divided into the grid of 100 smaller squares by strings attached across the frame. Most of them are a metre squared, but it's important to notice that not all of them are. Some of them are half a metre by half a metre, and therefore you have to times the length by the depth, which would give 0.25 metres squared. So quadrats are placed on the ground at different points within the area you're investigating, and the species frequency, i.e. how often a species is found, or the number of individuals of each species, is recorded in each quadrat. So the percentage cover of a species can also be measured by counting how much of a quadrat is covered by the species. So you count a square if it's more than half covered, but you don't count it if it's less than half. So percentage cover is a quick way to investigate populations, and you don't have to count all the individual organisms. Quadrats are useful for quickly investigating areas with plant species that are within a small quadrat. Areas with larger plants and trees need very large quadrats. So as an example here, species A, the blue one, you wouldn't count that one. Yes for that one, no for that one. Yes, 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 yes. These two, no. It's less than half. So that is 1, 2, 3, 4, 5, 6, 7. Maybe 8. And then 9, 10. And then species B, the green one, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Species C, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24. Now it's important to notice that I've drawn this quadrat, so the squares aren't even, but they should really be even 10 by 10 squares. So transects. You can use lines called transects to help find out how organisms are distributed across an area. So if you were showing how species change from one area to another, so this would be good for things like sand dunes, so to show succession. So at the very beginning of the sand dune, there would be very hardy, your pioneer species like your marram grass. And then as you get further up, you might get more grasses, you might start to get some shrubs, then you might start to get some small trees, then you might eventually end up with some large oak trees. So that is showing the change across an area. So for example, how plant species change from a hedge towards the middle of a field. So you need to know about belt transects, and this is where quadrats are placed next to each other along a transect to work out the species frequency and percentage cover along the transect. So you would place it down every single time next to each other. But then you can also get interrupted belt transects. These are what are most commonly we use. So instead of investigating the whole transect, you can measure using quadrat placed at regular intervals, for instance every 2 metres. So this makes it easier to cover a large distance. So every 2 metres you place your quadrat down and you take your measurements of percentage cover. Now if you wanted to capture motile organisms that are moving, you might need to use equipment to capture them. So the best method of capturing organisms will depend on what you're studying. So if it's a flying insect, then you'd probably use a sweep net. If it's an aquatic animal, you'd use a net. If it's a ground insect, you'd probably use a pitfall trap. So they are the different methods of studying populations in an ecosystem.